Bacterial cell wall and its associated structures. Importance of cell wall. It provides shape and strength to the bacteria and the protection from environment. It protects the cell from osmotic lysis. It protects the cell from toxic substances. In pathogens, contribute to pathogenicity. Provides classification of bacteria into gram-positive and gram-negative by gram staining. The teichoic acid present in the cell wall of gram-positive bacteria provides negative charge to the cell membrane, and that helps stabilize the overall membrane. Composition and structure of peptidoglycan. Peptidoglycan or murin is an enormous mesh-like polymer composed of many identical subunits. The polymer contains two sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid, and several different amino acids. The peptidoglycan layer in the bacterial cell wall is a crystal lattice structure formed from linear chains of two alternating amino sugars. The alternating sugars are connected by beta-1,4 glycosidic bond. The peptide chain of 4 alternating D and L amino acids is connected to carboxyl group of N-acetylmuramic acid. D amino acids are D-alanine and D-glutamic acid. L amino acids are L-alanine and L-lysine. In order to make a strong mesh-like, peptidoglycan subunits must be joined by cross-links between the peptides by glycine. Difference between gram-positive cell wall and gram-negative cell wall. Gram-positive cell wall is thick, made up of 90% peptidoglycan, contains teichoic acid, have low lipid content, outer membrane is absent, and thin periplasmic space. Gram-negative cell wall. The cell wall is thin, made up of 5 to 10% peptidoglycan, do not contain teichoic acid, very high in lipid content, contains outer membrane and thick periplasmic space. Outer membrane. It lies outside the thin peptidoglycan layer in gram-negative bacteria. Protects the inner membrane against several antibiotics, dyes, detergents, lysozyme, etc. Contain outer membrane proteins contributing to virulence, transport system, etc. Contains porins that are static water-filled transmembrane channels, allowing molecules to pass through them. Lipopolysaccharides. They are large, complex molecules consisting of lipid and a polysaccharide joined by covalent bond. They are found in the outer membrane of gram-negative bacteria, act as endotoxins, and elicit strong immune response. It is the major component of the outer membrane of gram-negative bacteria and protecting the membrane from certain kinds of chemical attack. It also provides negative charge to cell membrane and stabilizes overall membrane structure. Lipopolysaccharide is also known as exogenous pyrogen. It contains 3 parts: O antigen, core oligosaccharide chain, and lipid A. O antigen. The composition of O antigen, this repetitive glycan polymer, varies from strain to strain. They impart antigenic specificity. Core oligosaccharides. They attach directly to lipid A. Lipid A. It is a lipid component of an endotoxin held responsible for toxicity of gram-negative bacteria. May cause fever, diarrhea, and possible fatal endotoxic shock when released in circulation. Porin protein. They are present in the outer membrane of gram-negative bacteria. These are beta-barrel proteins that cross a cellular membrane and acts as a transmembrane channel, allowing the diffusion of molecules. Unlike other membrane proteins, porins are large enough to allow passive diffusion. Periplasmic space or periplasm is a space between the inner cytoplasmic membrane and external outer membrane of gram-negative bacteria, or equivalent space between the cell membrane and the cell wall in gram-positive bacteria. The space contains a loose network of murin or peptidoglycan chains, as well as gel containing hydrolytic and degenerative enzymes. Other enzymes are involved in various biochemical pathways, including peptidoglycan synthesis, electron transport, and alteration of substances that are toxic to the cell. In some species also contain beta-lactamase enzyme responsible for degrading penicillin. Teichoic acid. It is present in the cell wall of gram-positive bacteria. They are bacterial polysaccharides of glycerol phosphate or ribitol phosphate linked via phosphodiester bonds. The teichoic acid is covalently connected to either peptidoglycan or to plasma membrane lipids. In the latter case, they are called lipoteichoic acid. It acts as an antigen, being able to stimulate specific immune response. Slime layer or envelope. Many bacteria secrete extracellular polymer outside their cell walls, usually composed of polysaccharides and sometimes proteins. When the layer is well organized and not easily washed off, it is called a capsule. When it is a zone of diffuse unorganized material that is removed easily is called slime layer. It is composed of protein or glycoproteins. It may protect the cell against ion and pH fluctuations, osmotic stress, enzymes. It can promote cell adhesion to surface. It seems to protect some bacterial pathogens against the host defenses, thus contributing to their virulence. It can also be used as a food reserve for the cell. Thank you for watching. Don't forget to subscribe my channel and hit the notification bell to remain updated for more such videos.